Good afternoon and welcome. I'm joining you live from the Shinedown in Canberra, and I wish to acknowledge the Ngunnawal people who are the traditional owners of Canberra and surrounding areas. The Academy acknowledges and pays respect to the traditional owners and the elders past and present of all the lands on which we operate, live and work. They hold the memories, traditions, cultures and hopes of Aboriginal and Torres Strait Islander peoples of Australia. My name is Professor Malcolm Sandbridge, and I'm Vice President and Secretary of the Physical Sciences for the Australian Academy of Science. I'm thrilled to be here today for a special event to celebrate 5 of the Academy's career honorific awardees. These awards recognise lifelong achievement and outstanding contribution to science in Australia and internationally. We are delighted to be able to celebrate awardees from 2022 and 2023, and we look forward to hearing from them. So let's get started. To introduce our first awardee, we welcome former Academy Vice President and former Secretary for the Biological Sciences, Professor Helene Marsh.
Thank you. I'm delighted to be here today celebrating Academy Honorific Awardees. In particular, I join you today to celebrate the Suzanne Cory Medals for 2022 and 2023. The Suzanne Cory Medal is a career award that recognises outstanding research in the biological sciences. It honours the contributions made to science by Academy Fellow and past President, Professor Suzanne Cory, who, as a molecular biologist, has made major contributions to understanding the genetic causes of cancer. In 2022, this medal was awarded to Professor Georgia Chenevix-Trench. Before we hear from Georgia, I'll read her official citation. Professor Georgia Chenevix-Trench is a cancer geneticist interested in both inherited and acquired genetic variants that contribute to the risk and development of cancer. Her main focus is on breast and ovarian cancer, but she's also made major contributions to inherited skin and gastric cancers. In the last 15 years, her main focus has been on genome-wide association studies to identify inherited genetic variances associated with cancer risk. These have identified over 200 regions of the genome associated with breast cancer risk. This information is currently being used in international clinical trials to stratify women for breast cancer screening, but has also transformed our understanding of the biological basis of breast cancer. Georgia's main focus now is to identify the relevant susceptibility genes in these 200 regions to determine how they contribute to breast cancer risk and whether this information can be used to treat breast cancer or even to prevent it. Now let's hear from Georgia.
Thank you. I'm extremely honoured to receive this award, named after someone who's always been such a role model, particularly for women who want to combine a research career with motherhood. And I want to begin by pointing out the obvious, that everything I've achieved has been on the back of many wonderful students, postdocs, research assistants and colleagues. I've been told that I can talk about my entire career and current research, so I'll try to do both of these and set the scene with my earliest influences. My earliest memories are from Kenya. We then moved a long way to Tipperary, and I ended up at a school founded by my great-great-grandfather for the education of girls. He had 6 of them, and it was there that I was introduced by an excellent science teacher to the laws of Mendel. From there, I went on to Trinity College Dublin, which has always supported the education of women, so much so that there was a time at the start of the last century when women from Oxford and Cambridge travelled to Dublin to get their degrees. It was here that I met this remarkable man who persuaded me and many others that genetics was the way to go, and then guided me to a PhD scholarship in the US. I ended up at the Medical College of Virginia in Richmond, where it's probably fair to say that the only remarkable thing about my PhD is that I met this Australian and learned a lot about independence. But I also learned a lot about clinical genetics, which turned out to be very useful when I got to Brisbane. The independence also stood me in good stead when we got to QIMR in 1986. I always say that getting a research job in Brisbane in 1986 was like getting one in Vladivostok. But how things have changed. Brisbane certainly is no longer like Vladivostok. Thanks largely to Nick, Brisbane is now the centre for complex disease genetics in Australia, as also indicated by these Ruth Ghani awardees, all of whom are or were from Brisbane. My first successful project was finding the gene that causes a rare complex inherited disorder and thousands of skin cancers. I flew all around Australia from Cairns to Geraldton and more, diagnosing and collecting blood from families with this syndrome. I then mapped the gene responsible for chromosome 9, and with a group of collaborators, we identified mutations in the patched gene. This led to an explosion of interest in the patched pathway in cancer, for which there are now 3 approved drugs. To me, this exemplifies the dictum of William Bateson, to treasure your exceptions. What I really learned from this was that in genetics, resources are everything. You have to have the largest sample size you can. And it turns out that Australia is a very good place to acquire them. I moved into research on breast cancer, mostly because Joe Sandbrook had moved from the US to take Melbourne by storm. It was at a time when the first breast cancer gene had been identified, but no one in Australia had played any part because they hadn't collected any families. Joe started this consortium, CONFAB, which recruited families through the new family cancer clinic setting up. And we quickly became the best resource in the world for research into the familial aspects of breast and ovarian cancer, supporting over 200 research projects here and overseas. One of these research projects was mine, looking for additional breast cancer susceptibility genes in families like this. We lucked into finding this particular mutation in a gene called ATM and published this in 2002. And with a lot of additional work from around the world, by 2014, it was approved as one of the breast cancer susceptibility genes worth clinically testing for here and elsewhere in the world. However, mutations in genes like these only account for a small percentage of breast cancer cases, and not even all of the big breast cancer families. The really big advance in the last 20 years for those of us studying common complex diseases has been the advent of GWAS. The basic premise is very simple. You collect blood samples from thousands, now millions of cases with a disease or trait and controls without, and you measure hundreds of thousands of genetic markers scattered through the genome. And then you look for those that are significantly more common in cases than controls. The results are shown as a Manhattan plot, in which the markers are ordered along the x-axis by chromosome position, and the significance is plotted on the y-axis. Because of the multiple testing problem, a p-value of less than 5 times 10 to the minus 8 is typically accepted as the threshold for significance. As soon as the technology became available to measure so many genetic markers cheaply, we started 2 international breast cancer consortia, because it was obvious from the start that we'd need large numbers of samples. Doug Easton from Cambridge set up BCAC to look in breast cancer cases and controls in the general population, while I started SIMBA because I was interested in a slightly different question, i.e. whether the same variants we found in BCAC could affect the breast cancer risk in BRCA1 and 2 mutation carriers. Although these genes are regarded as high risk, not everyone who carries a mutation develops breast cancer. These 2 consortia have run in parallel now for almost 20 years, and now each have at least 100 participating sites from all over the world, and we are genotyping hundreds of thousands of samples. We found over 250 regions of the genome associated with breast cancer risk in the general population, and usually also in mutation carriers. And expect this to double next year because we've doubled our sample size. Apart from the fascination that you can find the genetic basis of everything from nail biting to cardiovascular risk, there are 2 major applications of GWAS. One is that although each of the risk variants has a very subtle effect, in combination their effects can be striking. This was shown initially by Peter Fisher and Naomi Ray, who transferred the concept of polygenic risk scores from cattle to humans. I illustrate this PRS by showing the effect of a high or low PRS in women and men who carry a BRCA2 mutation. You can see that in women, the risk of breast cancer to age 80 varies from 48 to 86% for women at the 5th and 95th percentile, a big difference that we think might help women to decide when they have prophylactic mastectomies, or indeed whether to. We're now doing a clinical trial in Australia to find out whether giving this information to mutation carriers changes their behaviour and saves lives. In men, the difference in prostate cancer risk, BRCA2 is also a prostate cancer risk gene, is similarly striking, although the only recommendation here would be for more intensive and frequent screening. But it does pave the way for clinical trials targeted at high-risk men. Another major application is to repurpose or design new drugs for complex traits. But first we need to understand what these risk variants do, and specifically what genes they target. Here the field found the first problem is this double-edged sword. Genetic variants are not all transmitted independently through generations, but because of evolutionary history, they can be inherited in disjointed blocks called linkage disequilibrium. All these brown nucleotides are clearly always inherited together. The good news is that in this case, we can just measure the A nucleotide here in the red box, and if associated with breast cancer, we can infer that all the other brown nucleotides will be associated. The problem is that probably only one of them is causal, and this could be this variant down here in the blue box. The other problem is that even when you've narrowed down the likely causal variants by fine mapping, working out what they do isn't straightforward. They rarely change a protein, but instead they are in regulatory elements called enhancers. But DNA is not arranged in a linear fashion, but loops extensively so that enhancers can be brought into proximity with genes some distance away. In this schematic, the red enhancer regulates the expression of genes 1 to 3, but not gene 4. So the last problem is whether genes 1, 2, and 3 all influence the trait, or just one of them, and if so, which one? So you can try to work out the target genes, one region at a time, as I and my colleagues used to. Sometimes you find genes with known or suspected roles in breast cancer, such as TERT, which is responsible for maintaining the ends of chromosomes, but often you identify genes that were never previously known to play a role, opening our eyes to how breast cancer really develops. The reason that this is so important is that it is now well known that the likelihood of getting a drug to market doubles if it's targeting a protein encoded by a gene implicated in risk through genetic studies. Doubling the chance of success is economically huge, but even so, it's extraordinarily expensive to develop new drugs. So there's a big emphasis on repurposing drugs developed for one indication to another disease. In my lab, we're working on a drug, Chenevix-Trench, which was developed for sickle cell anemia. It isn't effective for sickle cell anemia, but the clinical trials have shown no side effects from long-term use. You can see here that with my colleague Cara Britt, we've shown that Chenevix-Trench treatment can delay or even prevent mammary tumors in mice. So our long-term aim is to see if this can be used in humans, particularly those whose polygenic risk score shows them to be at high risk. So back to the issue of what the new breast cancer risk genes do. This figure illustrates what are known as the hallmarks of cancer. We think and have shown that the breast cancer risk genes could act in any pathway. For example, caspase-8 plays a role in avoiding immune destruction, according to our most recent analyses. We're never going to find out the target genes of all 250 risk loci by studying them one at a time. So instead, we've moved to screens using CRISPR technology, where you can examine thousands of genes at once by knocking them down or activating them, and then looking at the effect on function. So we're using pooled CRISPR screens in which you aim to get a single guide RNA into each cell, and then measure the abundance of these guides after applying a selective pressure. We've done this so far with considerable success to find genes involved in proliferation in vitro, tumorigenicity, DNA repair, and T-cell killing. I would therefore like to conclude by saying that I think this is really the only way forward for finding genes for complex diseases in order to capitalize on the enormous success of GWAS, and to thank you for your attention.
Now it's my pleasure to welcome live with us today Professor Georgia Chenevix-Trench. Thank you, Georgia, for joining us, and congratulations on this award.
Thank you very much.
Now we'll go to some questions. I have one here to kick us off. So if you're interested in seeing if you have these mutations, what sort of processes do you need to complete to find out? Who should get tested for the likelihood of carrying these mutations?
Well, in Australia and in other places, testing is mainly based on family history. If you've got multiple family members, particularly affected at a young age with breast or indeed ovarian cancer, you're likely to get tested. Ethnicity plays a part. Some of these mutations are much more common in the Ashkenazi Jewish population, of course. And in that case, family history might be missing, but simply being an Ashkenazi Jew will get you tested. Sex is also important. Male breast cancer is associated with CA2 mutation, so a family member with male breast cancer. And then age, getting the cancers young, puts you in a high-risk group, and pathology. So there are particular kinds of breast cancer that are more common if you carry these mutations. So if you fulfill one of many different criteria, then you probably need a referral from your GP to a family cancer clinic. And these days they test you not for one gene, but for a whole panel of genes. But there are also some direct-to-consumer opportunities where people just go straight to a commercial company for testing, but don't get such associated counselling. And I think we're moving gradually towards population screening. In Israel, where the mutation rates are high, they're starting population screening, so every woman gets tested.
Right, thank you. Can I ask, with these GWAS, how many samples do you need? And I believe it's large, so why so many?
Well, that depends a lot on the trait. So by enormous luck, the first GWAS that was published, at least, was for macular degeneration, and they only had about 100 cases. But luckily, there's one really strong gene for macular degeneration, and they found it. If they'd started with something like depression, where there are many genes, each of tiny effect, this whole revolution might not have happened, because they certainly wouldn't have found anything with 100 cases. So it really depends on what we call the genetic architecture. For some diseases, you probably need tens or even hundreds of thousands because all the genes have a tiny effect. There are others where there might be a reasonably big effect with an odds ratio of 1.5 or so, and then you can get away with maybe 1,000 cases of controls. Initially, 1,000 cases of controls was regarded as a sort of minimum, but I think these days it would be much more than that. Of course, it begs the question what you do if you've got a really rare disease. You know, if you just can't get 10,000, you start with 1,000 or do you not bother? And I think the answer is you start with 1,000 and see if you find anything.
That's interesting. So let me ask the opposite question. If you don't have any of these genetic mutations and say live a healthy lifestyle, can you still develop breast cancer?
Yes, absolutely. I mean, first of all, we know we haven't found all the variants yet, so you could be missing all of these, but still have some others. But we also know they're major environmental risk factors. One of the most common is the number of children you have. Breast cancer rates have increased quite dramatically in the last 50 years or so as family sizes go down. How long you breastfeed for, when you have your first child, all of these can influence your risk. Many of those themselves have a genetic basis, but we think of things like how many children you have as an environmental risk factor. But absolutely, you can develop breast cancer without these particular variants or mutations.
Right. Okay. And can you use these GWAS to find mutations in a population for any trait?
Any trait that has a genetic, at least a partially genetic basis, and there's almost no trait that doesn't. So my husband was challenged once to find something with no genetic basis. So he said he would look at church attendance. And in fact, church attendance has quite a genetic basis. Not which church you go to, but whether you go to church. So the diversity of traits where people have done these GWAS is completely extraordinary. Going, how much cheese you eat, whether you bite your nails, whether you're an affable person, you name it, you can do GWAS for anything.
Wow, I didn't know that. So how did you come by the idea that you mentioned in your talk that the drug Chenevix Chenevix, I think it was, which isn't effective, as you said, for sickle cell anemia, might be effective in helping to delay and prevent mammary tumours. How did you,
I should have made
make that leap?
Yeah, I skipped over that. The variants of this locus are right within a gene called KCNN4. So in this case, it was pretty obvious to us that KCNN4 itself was the gene that's being targeted, and snakeapop is an inhibitor of KCNN4. That's why it was developed. But one of the important steps is you have to work out whether an increased level of KCNN4 is associated with risk or a decreased level. Because obviously you want to use an inhibitor only if increased levels are associated with risk. And so we were able to show that independently. If you carry the risk variant at this locus, you have more KCNN4, and therefore inhibiting it seemed like a good idea. And it turns out to be so.
That's interesting. So let me ask, if a mutation in the ATM gene and I think it was the BRAC gene increases the likelihood of developing breast cancer, what is the function of these in genes in our body?
Well, all of those, in fact, just about all of what we call the high-risk genes that we found are involved in some way in DNA repair. So ATM itself particularly senses DNA damage. BRCA1 and BRCA2 are involved in that repair. So the cell makes millions of errors with every round of replication. And in a well-functioning cell, those are rapidly repaired by ATM and these other genes. But if they're mutant, then you accumulate DNA damage, which ends up developing into a breast cancer or another tumor.
Okay. So I think many of us have heard from the media. I was under the impression that if you had a mutation in the BRCA1 or BRCA2 gene, you were most susceptible to breast cancer. But from your last slide about the hallmarks of cancer, you show that there are a lot more target risk genes than BRCA. So has this information changed how people get screened for mutations that lead to breast cancer?
No, because the last slide really related to the genes we found through GWAS, and the individual effect of each of those genes is tiny. The combined effect in a polygenic risk score can be quite big, but nobody would test for variational mutations in the individual genes because their effects are so tiny. So we have this sort of distinction between, the sledgehammer genes like BRCA1, 2, ATM, and about another 6 or 7, which are now routinely tested for clinically, versus all the others we found through GWAS that can act through those different hallmarks of cancer that are biologically really interesting, that might lead to the development of a new drug or the repositioning of a drug, but are not valuable to test for clinically.
Okay. So we can go to some questions from the audience. The audience can put them in on the screen, and I'll monitor that as we go on. I don't see any at the moment, but let me ask, I know you mentioned a p-value of 10 to the minus 8 was required. I mean, I teach a bit of statistics and I think, wow, that's small. But is that normal in these sorts of studies? You really need that sort of strong evidence.
And that's why you need so many samples. And it's because we're testing at least 300,000 markers at a time, and then often inferring 1 million or so markers. So there's this huge multiple testing problem. And so people have been really quite vigorous in maintaining that as a threshold. I mean, obviously, in one study, you might get a p-value of 10 to the minus 7, and it's not accepted as real. But if you do a further study and you find, the same effect, you can combine them and cross that threshold. But that was something that was really put in place very early on. And I think is one reason the field has been so successful, because people have been rigorous in their claims. Whereas previously, when we were studying single genes and using a p-value of 0.05, hardly anything ever replicated. There was big as publication bias, and we just weren't rigorous enough. So I think we learned from that experience, and GWAS have done it much better.
And it's practical to do that. Perhaps it wasn't in the past in terms of the experimentation.
It is practical, and it's practical for two reasons. One is that for the sort of 20 years before this revolution, a lot of people were collecting samples and then studying single genes. So we had a lot of samples already collected. So that was fantastic. And also because the technology used to do this is relatively cheap. So these days you can measure 300,000 markers for $50. So, it's not within a regular NH&MRC budget, But, you know, it's certainly feasible to do at the sort of scale that we do through collaborations and foreign budgets, etc.
That's wonderful. Okay, well, thank you again. I think that's about all we have time for today. So I'd like to thank you and congratulations again, Georgia.
Thank you.
Thank you.
Bye.
Bye. So now we move on to introduce our next awardee. We welcome back Professor Helene Marsh.
Thank you. As I mentioned earlier, the Suzanne Cory Medal is a career award that recognises outstanding research in the biological sciences. It honours the contributions made to science by Academy Fellow and former President, Professor Suzanne Cory. In 2023, this medal was awarded jointly to Professor Cath Lovelock and Professor Terry Hughes. We're now hear from Professor Terry Hughes, but before we do, I'll read his official citation. Professor Terry Hughes has made a superlative and sustained contribution to marine biology and science leadership in Australia and globally. Terry's early research pioneered new understanding of the population dynamics and life histories of corals and of the ecology of coral reef ecosystems. Among his most significant research has been his groundbreaking exploration of the resilience of coral reefs to pollution, overfishing, and climate change, and on the dynamics of tipping points and regime shifts. Throughout his distinguished career, Terry's research has provided innovative and practical solutions for improving coral reef management and governance. He's also a highly cited researcher with many publications in science and nature, and he's been the founding director and driving force between the ARC Centre of Excellence for Coral Reef Studies, providing leadership and mentoring for a large team of researchers of all career stages. Now let's hear from Terry.
Well, thank you very much, everyone. It's a great honour here to be today for the Susan Cory Medal Award talk. I feel very honoured and very privileged, and thank you, Helene, and thank you, Malcolm. I'm going to talk today about coral reefs in the Anthropocene, meaning the modern period that we've now entered. I've been very privileged to have studied coral reefs throughout my career. Coral reefs are really important, not just for their iconic beauty and biodiversity, but also from a very practical standpoint. They support the livelihoods, food security and well-being of hundreds of millions of people throughout the tropics and subtropics. This is a map of the world showing the location of the world's coral reefs. I began my studies as a PhD student in the Caribbean. I took this photograph all the way back in 1979. It shows two species of three-dimensional branching corals that are now considered to be so rare that they are at risk of extinction. But back when I was a student, they were incredibly abundant. So one of my earlier research projects documented the rapid decline of many Caribbean coral reefs. This is a photograph showing highly abundant branching corals. And if you look at that same metal pole that's driven into the reef, this is the same location a few decades later, and you can see it's obviously being degraded. If we look at some data on that, the top graph is the percent coral cover on this axis against year at different depths, and we see this rapid decline over a period of less than 20 years, and a coincidental increase in the amount of seaweed or macroalgae. And basically this reef has collapsed. We call that a regime shift from a coral state to a macroalgal state. And importantly, this transition occurred even before mass coral bleaching, which is a new modern phenomena, which is superimposed on the older stresses to coral reefs that haven't gone away, namely pollution and overfishing. So today, the dynamics of the world's coral reefs is highly influenced by global warming, or more correctly, anthropogenic heating. This is a tweet I put out a few days ago showing the bleaching forecast by NOAA for the Caribbean basin. And basically red here is bad. They're projecting in this northern summer 100% probability of severe bleaching occurring. And it's already occurring through the month of July. That's earlier than any year before. And temperatures in the sea won't peak until early September. So it's looking pretty bad for the Caribbean basin this northern summer. And we're very concerned about what might happen to the Great Barrier Reef in 6 months' time, particularly if the developing El Nino continues to strengthen. The Great Barrier Reef has now bleached 6 times since 1998. 1998 and 2016 were strong El Nino years, but we no longer need an El Nino to trigger mass coral bleaching. And in fact, we saw bleaching during cooler La Nina phases of ENSO cycles for the first time in 2022. We're likely to see the 7th coral bleaching event early next year on the Great Barrier Reef. Some of you may be familiar with what bleaching looks like, others may not. So I've included this very short video. It doesn't have any sound, so I can explain what's going on. That's me looking at the window of a small airplane. So one of the major questions about bleaching events is where are they occurring, where are they the most severe, and why are they the most severe where that's happening. This is what bleaching looks like from a helicopter or a small plane. And so we can map out the extent and severity of coral bleaching. We can compare those maps of bleaching with maps of heat exposure and temperatures, which we get from satellite data provided by BOM and NOAA. This is what bleaching looks like underwater. You can see the corals have turned white because they've lost their symbiotic algae, which gives them most of their color. That coral there is already dying. So in 2016, when this video was taken, we saw on average a 30% loss of corals along the full extent of the Great Barrier Reef, with much higher mortality rates further north, for the northern third of the Great Barrier Reef. And I'll show you a graph of that in a moment. We've now seen 6 of these bleaching events, and we look at the cumulative footprint of the last ones in 2016, 2017, 2020, and 22, we get this map like this. And the point here is that we're running out of places that haven't yet bleached, which are shown here in dark blue. So 80% of the Great Barrier Reef has bleached severely since 2016, enough to cause at least some coral mortality. And only 2% of the Great Barrier Reef now has shown no level of bleaching since 2016. This is a map of the mortality that we measured underwater by going to 168 reefs at the time of bleaching in 2016, and again 6 months later in October, November. Red here means more than 50% mortality. Green means 0 mortality. And if we look at the satellite data using a metric called degree heating weeks, which measures the level of thermal stress, how long the water was hot, and you can see that the hottest water was in the northern third of the Great Barrier Reef, the coolest was in the south. So temperature and the duration of spikes of extreme temperature explains both bleaching and the loss of corals. I'm often asked the question, will we still have a Great Barrier Reef in 50 years' time? And my answer is a cautious yes. But it really is up to us. It depends on where temperatures go from now. Ideally, what we need is for global temperatures to equilibrate as soon as possible and as low as possible. We're all familiar with the 1.5 to 2 degree global average warming target of the Paris Agreement. 1.5 degrees, I think, is very doable for the world's coral reefs, although the transition that we're already seeing will continue. 2 degrees will be very difficult. And I think if we still have business as usual emissions in 20 years' time, then we will be looking at very degraded reefs worldwide. So we're on a trajectory. That trajectory has begun already. The mix of coral species is responding because some species are more susceptible to thermal stress than others, and some are also much better at bouncing up back in the gaps between one bleaching event and the next. The problem, of course, is that the gaps between one event and the next continue to shrink. In conclusion, I think recurrent bleaching has already changed reefs forever. The barrier reef today is very different from the reef of 30 years ago, and it will be very different again in 30 years' time. Restoration, unfortunately, is not the answer. We're now seeing in Florida, where there is extreme heat exposure this northern hemisphere summer, many of the coral nurseries have been destroyed by the spike in temperature this summer. What is the answer is reducing emissions and local drivers like pollution and overfishing, and there's no time to lose, and particularly in Australia, for reducing greenhouse gas emissions to secure a future for the world's coral reefs. Thank you so much, and I look forward to answering some questions.
And now it's my pleasure to welcome with us live Professor Terry Hughes. Welcome, Terry, and congratulations on this award.
Thanks very much, Malcolm. It's a pleasure to be here.
And I think I'll kick off with some questions, if I may.
Sure.
So can I ask, you mentioned bleaching and you showed a lot of data about the degree of bleaching. So what actually happens to corals when they become bleached? And is it possible for them to recover from bleaching, or is there some threshold where they won't recover?
Bleaching is a stress response. Known about it for a long time, because if you take a coral from a reef into an aquarium and torture it in various ways, it will usually bleach if you make the water too hot or too cold. If you add sediment or some sort of pollutant, you can measure the impact of those changes in an aquarium in the laboratory. But what we're seeing now at a regional and even a pan-tropical scale is something very different. And that's the impacts of global warming. Higher temperatures, particularly spikes of temperatures during heat waves, are causing unprecedented large-scale bleaching, which can be very destructive.
As the climate warms and the water warms, is it possible for the Great Barrier Reef to move south? Is it possible to grow south as the climate and the conditions change?
The issue there is one of time scale. So already in the last couple of decades, we are seeing individual species that live in the tropics moving either towards the north in the northern hemisphere or to the south. So in places like the Ryukyus chain, south of Japan, we're now seeing species that are tropical showing up in the southern edge of the main islands of Japan. In Sydney, we're seeing tropical species overwintering for the first time in recent years. So we are seeing that expansion of geographic ranges of individual species, but it's going to take a very long time indeed for the Great Barrier Reef as a geological structure to expand further south. So that's a very different time frame from the expansion of individual species that we're already seeing.
Yeah, different timescales. Okay. And what are the ramifications of the Great Barrier Reef if it completely dies? What are the regional and global ramifications?
Well, ecologists like me rarely talk about an ecosystem dying. Ecosystems don't have birth rates and death rates the way a population does. We rather talk about ecosystems changing. You can use terms like degraded or collapsed, which can sometimes be a little bit judgmental. So I don't think the Great Barrier Reef is going to, quote unquote, die. Nature abhors a vacuum. So even at extreme levels of future warming, there will be a tropical ecosystem off the northeast coast of Australia. Will likely have far fewer corals and probably more sponges and seaweed and other things instead of corals if we get to extreme levels of warming. But to be honest, if we get to temperatures that are so high that the Great Barrier Reef becomes unrecognizable, it will be the least of our problems, because we'll have huge issues with agricultural production, with sea level rise, and so on. So that's one of the reasons I'm actually fairly optimistic that we will be able to save the world's coral reefs and take action on rising temperatures, because it's starting to get very uncomfortable, not just for coral reefs, but for human society.
Yep. Okay. And you mentioned in your presentation there 2016 as a date of study, detailed study, and you mentioned the change from 30 years ago. So do we know when the reef actually started declining? Is that possible to even know?
Yes, it is. So we have historical records on things like fishing, particularly after the arrival of Europeans into Australia. We also saw huge changes in land use. So we know, for example, that the amount of sediment and nutrients running off the coast into the inner part of the Great Barrier Reef has increased by about 5-fold in the last 200 years. So yes, the Great Barrier Reef today is an altered ecosystem. For the large part, it's still in reasonable nick. But the insidious thing about global warming is there's nowhere to hide from it. Even the most pristine, best managed, most remote place where there's almost no fishing or no runoff of pollutants from land, even those very isolated locations are nonetheless vulnerable to rising sea temperatures.
I see. So on the other side of the coin, can you tell us whether there are any healthy reefs today in Australia or the world?
Healthy is another term like dying ecosystems. It's not really an appropriate word to describe the condition of an ecosystem. We don't really talk about sick ecosystems. We talk about altered ecosystems, or sometimes degraded ecosystems, where the characteristic that we measure is the shift in the composition, in the mix of species and the proportions of different species. So using that metric, the Great Barrier Reef, and in fact, all the world's coral reefs, are different today than they were 30 or 100 years ago. They have begun a transition, which is largely pushed along by rising sea temperatures. What we really need to see happening, as I mentioned in my talk, is for that rise in temperatures to equilibrate. And hopefully it's something close to the 1.5 to 2 degree targets of the Paris Agreement. And then we'll have a period of readjustment. So I'm talking about the trajectory that the Great Barrier Reef and other reefs are already underway, that will take another half century to equilibrate. And then I think once we've run that gauntlet of huge impacts currently of global warming, hopefully we'll see a stabilization and a rebuilding of a reef, a different reef, but one that is still recognizable and still functional as a coral reef.
Okay. So in terms of triggering of coral bleaching, the question is, what triggered the 2022 Great Barrier Reef bleaching if there wasn't an El Nino weather pattern during that year? Do we know that?
Yeah, so if we look at El Ninos, they happen on an irregular cycle about every 5 years or so. And we've had many of them going back to the end of the last ice age. But it wasn't until 1998 that El Ninos became dangerous for coral reefs because of the underlying rise in temperature due to global warming. Fast forward another couple of decades, we no longer need an El Nino to trigger a bleaching event. We just need a record-breaking hot temperature, and that's what we had in 2022, despite it being a relatively cooler period of ensocycles, namely La Nina. So the climate modelers are predicting that we could see annual bleaching, back-to-back bleaching in 2 consecutive years. Every year from mid-century, we've already seen back-to-back bleaching in 2 consecutive years on the Great Barrier Reef in 2016 and 2017. So El Ninos don't cause bleaching. Warming temperatures due to burning fossil fuels and changes in land use are the root causes of coral bleaching.
I see. Okay. And you said in there that there was a regime shift from corals to microalgae. So why can microalgae survive in these new environments so much better than coral? And For example, could the aquatic life currently living in coral reefs shift to living in microalgae?
Yeah, so it's macroalgae, so they're big stuff, not microalgae.
Sorry, macroalgae. Sorry.
Yeah, they're sometimes called fleshy seaweed. So they're typically 10 centimeters or maybe 30 centimeters in height. They do provide some three-dimensional structure, but you have a very different suite of species that live in and amongst seaweed compared to what lives in and among coral. So if we look at fish, for example, the reason why loss of coral cover matters is because corals are the architect. They provide the habitat. The nooks and crannies in between branching corals in particular support many species. Some species eat coral. Many larval fish that are commercially important need live coral cover as a nursery for juvenile fish. So if you lose corals, you change the mix of species across the entire ecosystem because they're so important. So if we were to replace corals with algae, we'd have a very different system with different kinds of species, probably much smaller bodied fish. It would be a very degraded ecosystem.
Okay, thank you. We have a viewer online question that says, how can the public best advocate for coral reefs? And you yourself, how do you overcome climate fatigue working in the field?
Well, I think the good news is many people are aware now of coral bleaching. Coral reefs are sometimes called the canary in the coal mine. It's not a phrase I use because I actually think coral reefs are not any more susceptible to global warming than many other ecosystems, kelp beds, for instance, mangroves, seagrass beds. All of those other marine ecosystems are quite possibly equally vulnerable to global warming. Coral reefs are certainly the poster child. So I think public awareness of the impact of bleaching is now very high, and I think increasingly the public is also on board in terms of trying to meet the goals of the Paris Agreement.
Oh, look, I very much enjoyed that, Terry. Thank you very much. I think that's all we have time for today, but let me thank you again and congratulations on this award.
Thanks so much, Malcolm.
Thank you. So now, to introduce our next awardee, we welcome back Professor Helene Marsh.
Thank you. As I mentioned earlier, the Suzanne Cory Medal is a career award that recognises outstanding research in the biological sciences. It honours the contributions made to science by Academy Fellow Professor Suzanne Cory. And in 2023, this medal was awarded jointly to Professor Cath Lovelock and Professor Terry Hughes. We'll now hear from Professor Cath Lovelock, but before we do, I'll read her official citation. Professor Catherine Lovelock is a leading global expert on the impacts of climate change on coastal wetlands and the role of coastal ecosystems in mitigating climate change. Her research demonstrates the important role coastal wetlands, mangroves, salt marshes, and seagrass play in mitigating climate change. Achieved by assimilating atmospheric carbon within living wetland plants, a proportion of this plant material is stored in sediments for long periods and is known as blue carbon. Cath has been pivotal in driving international research and policy regarding blue carbon and was instrumental in developing a voluntary blue carbon market in Australia that will play a central role in Australia's efforts to adapt to and mitigate the effect of climate change on Australia's coasts. Her research emphasises the important role of coastal wetland plants in accumulating substrates, a process that's particularly important in a changing climate where sea level rise will increase erosion and inundation frequency along shorelines. Despite this important role, Cath has cautioned that the capacity of coastal wetlands to adjust to climate change will become increasingly limited throughout this century unless planning decisions reduce pressures and facilitate landward retreat. Now let's hear from Cath.
It's my absolute pleasure to be here today as a recipient of the Suzanne Cory Medal for 2023. I'm sitting in Brisbane, so I'd like to acknowledge the traditional owners of the land in which I teach, do research, work, and that is the Turrbal and Jagera people, and I'd like to pay my respects to their elders past, present and emerging. Today, I want to talk to you about blue carbon, and particularly I want to talk about how blue carbon is more than just climate change mitigation. So we know that we have two huge challenges that we're facing, and that is that of biodiversity loss and also the threats of climate change. So in the international discussions on this, whether they occur at the Convention on Biological Diversity or at the UN when we talk about the decade of ecosystem restoration, or when there's been discussion of the Sustainable Development Goals, it's agreed that the key actions are the conservation and restoration of natural ecosystems. So the natural ecosystems that I've worked on in my career are coastal wetlands. And coastal wetlands include seagrasses, which occur on the lit waters, the sunlit waters of the shelf of Australia and elsewhere in the world, mangrove forests that are most developed in the subtropics and tropics, the salt marshes, which occur around Australia in an amazing array of different forms, supratidal forests, which are those that are connected to these other ecosystems, and they often experience extreme high tides and storm surges. And then in the arid zone, the vast high intertidal areas of sparsely vegetated salt marshes and salt flats. So all of these ecosystems have in common their importance to biodiversity. So this is a small clip of the kinds of animals and plants that you see in the mangroves of the Exmouth Gulf. And this is just indicative of the wide range of biodiversity that occurs in all of our coastal wetlands. So in this graphic, you can see the kinds of ecosystem services. So this is the benefits that people get from coastal wetlands. It focuses on mangroves, but the same can be said for all of those other types of coastal wetlands. They're important for fisheries because they are fish nurseries, fish graze in them, and they reproduce, so they're important in reproductive cycles. They're important for timber and fuel in some societies where livelihoods are linked to coastal wetlands. They're important for coastal protection because they slow floodwaters and they are a barrier against storms and storm surges. They're important for tourism. They're important culturally. People like to watch birds and go fishing and recreate in coastal wetlands. And finally, well, not finally, but they are also important for water purification. So all of that structure you can see in that picture, the roots, and they are important in trapping sediments and nutrients. And that means they're really good for those adjacent systems because they improve water quality. So, for example, the coastal wetlands are really important for the water quality of the Great Barrier Reef. And finally, we get to climate change regulation or climate regulation. And these ecosystems are important because they're vegetated, they absorb carbon dioxide from the atmosphere during photosynthesis, they grow wood. You can see the biomass in this mangrove forest, that stores carbon. And probably the thing that most differentiates them from other ecosystems is that the roots that they produce accumulate in the soil over time, because while they die, decomposition is very, very slow, because these soils are flooded, so they're very wet and they're salty, which depresses rates of decomposition. And that means soils grow in volume, they accumulate all this organic matter through time, and we have these large carbon stocks, blue carbon stocks, that can persist for thousands of years. But unfortunately, these ecosystems have been degraded, and that means they've become, when they're degraded, they become sources of emissions that actually give off CO2 as their biomass and soils are subject to decomposition. So this is a picture of loss of mangroves in Madagascar, where they've been harvested for timber. And the picture on the right is a picture of aquaculture developments in Indonesia, and throughout Southeast Asia, mangroves have been converted to aquaculture and also rice production. And 44% of the mangroves of Southeast Asia have actually been lost since the 1970s. Although those pictures are from degradation of those coastal ecosystems overseas, in Australia we also have large areas of coastal wetlands that have been converted for coastal development and that have been drained and converted to agriculture. For example, on the left, you can see drained wetlands that have been drained for sugarcane production. While on the right, you can see wetlands that have been cut off from the tide by levee banks so that fresh water accumulates on the landward side and can be used to grow pasture that feeds cattle. So there's an enormous amount of pressure on coastal ecosystems, and there also exists the potential to restore them, to restore their carbon sequestration capacity, and to avoid the emissions that are occurring as they remain in a degraded state. So how do we take this science, what we know about coastal ecosystems, how great they are for us, how they can sequester carbon, how they can protect us from storms, and turn that into action? Well, we have to actually cross this science policy chasm that often exists. And the importance of doing that is that strong policy indicates a strong commitment to conservation and restoration of these ecosystems. It reduces uncertainty and it really encourages action. So through my career, I've had the opportunity to be involved in both international and national policies around blue carbon. So prior to 2013, the IPCC did not provide any guidance on how countries should account for both the emissions associated with coastal wetland degradation or the removals of CO2 from the atmosphere that are associated with their functioning and their restoration. But in 2009, a blue carbon document was produced that coined the words blue carbon, the phrase blue carbon, that urged the recognition of marine ecosystems for their role in regulating CO2 in the atmosphere. This is a really important breakthrough and an important action to drive the international community towards recognizing the importance of these ecosystems and how it was important to conserve and restore them. In 2013, the IPCC offered guidance for the first time for coastal wetlands. So that's the document in brown. In 2015, the Australian government introduced coastal wetlands into its national greenhouse gas inventory. And why that's important is because that enhances the transparency of what is happening to our coastal wetlands over time. And flowing from that, in 2022, I was part of a science team that developed a blue carbon method for the Australian carbon market. And this allows proponents or projects to restore coastal wetlands and obtain a carbon credit, which they can then sell to fund further restoration activity. And the idea is that this method would enhance the capacity of Australian coasts to sequester carbon through their restoration. So this is what this blue carbon method looks like. It's essentially a recipe for developing a project and restoring coastal wetlands for their carbon value. So this is the blue carbon method. And we can look at the kind of project that we might do. And that is exemplified here by a project called the Blue Heart, which is being led by the Sunshine Coast Council. So they have a large area of relatively flood-prone land, some of which is unproductive for sugarcane, and that was drained wetlands before they were drained for sugarcane. So the plan is to restore those to what they were in the past, and in the process, if you look at that picture on the right, store carbon in the biomass and in the soils. So there are other projects in Australia also under development. So what we don't have that we do need is a way to characterise and reward landholders and other project proponents for blue carbon for their impacts, their positive impacts on biodiversity. And this photo is actually quite hard to interpret, but we're looking down on a herd of dugongs grazing in seagrass meadows. So what I'm looking forward to is methods that not only account for the carbon that's stored in a restored seagrass meadow, but also the benefit it provides to us by supporting biodiversity like dugongs. So one of the other projects that I have in my mind, this is a new passion for me, is to be working on the development of a method to reduce the impacts of feral animals on wetlands, both on the blue ones, those that are in the intertidal zone influenced by tides, but also the connected freshwater wetlands of floodplains. If you look at this video, you can see the damage that pigs have done to the floodplain of the South Alligator River in the Northern Territory. So we're working with Indigenous organisations and a team of scientists to develop a method, a carbon method, that hopefully will sustainably fund the management of feral animals on country by both reducing greenhouse gas emissions, but also enhancing biodiversity. So in conclusion, I hope I've convinced you that blue carbon can deliver for climate change mitigation, but it's also important for climate change adaptation and for biodiversity. I think as a cautionary note, though, we need to understand that the science is becoming stronger, that the negative impacts of climate change are beginning to take their toll on coastal wetlands. And I think in addition to doing wonderful work on blue carbon, that we also need from the government a stronger commitment to reduce emissions so that our projects of conservation and restoration of coastal wetlands and other ecosystems have a chance of withstanding future conditions. So with that, I'd like to provide my acknowledgements, and there's many of them, because it really takes a lot of collaboration and cohesive work by a whole range of actors to make things like blue carbon happen, to be implemented for the landscape. And I'm super happy to have been able to share this short story on blue carbon with you. Thank you.
And now it's my absolute pleasure to welcome Cath Lovelock with us live on the feed. Welcome, Cath, and congratulations to you for this award.
Thank you very much, Malcolm. Thanks for having me here.
So just a reminder to our viewers that you can ask Cath and others today a question using the QR code that's been on your screen. Just follow that and you can feed me a question to ask Professor Lovelock. So, Cath, what processes must happen to restore a carbon wetland that was once agricultural land?
Now, that seems like a really simple thing to do, but it's actually quite a difficult problem. So, you know, I think a lot of people at the moment are trying to work out how we go from understanding that it's possible, and we know that it's possible because people do restoration projects, but how do we do it at the scale we need to do? So we need to engage landholders, we need to engage the community, we need to engage industry, sort of take this up. And basically, you know, one of the things we need to do for farmers is to demonstrate that this is a financially viable option for them. So, you know, I don't think anybody's thinking about giving up, you know, agricultural production on the floodplains. But what we are thinking about is that those unproductive paddocks that are close to the coast, that are, you know, on the edge of the creek, you know, maybe they're not performing so well, and perhaps that is the, you know, the target for wetland restoration, particularly in the Great Barrier Reef catchments, where I think it's so important for water quality.
Okay, thank you. You mentioned Blue Heart in your video there. Can you tell me, how does that actually transform old sugarcane into blue carbon sinks? How does that work?
Well, the Blue Heart is a really interesting project. It's led by the Sunshine Coast Council, who have recognised that their land, you know, there's 5,000 hectares in the heart of the Sunshine Coast that is extremely flood prone, and they know it's going to be badly affected by sea level rise. So they've been, along with partners like Unity Water, purchasing land over time, and they're trying to engage landholders now as well to reconvert those lands into coastal wetlands. And that means lifting tidal gates or opening tidal gates, re-engineering the work that has been done to make them into sugarcane fields, and, you know, like modifying that water infrastructure or the drainage infrastructure to facilitate wetland restoration or wetland growth on those low-lying lands. And they know that what this is going to do for the region is, one, enhance tourism, because sugarcane has, as an industry in that area, has declined because the mill has closed, and really provide the flood protection. The flood protection is really at the heart of this for the rest of the Sunshine Coast. So it's sort of like, we're going to create a wetland that's going to do this coastal protection function, and it's going to be a carbon sink.
Okay, thanks. So let me ask, are there wetlands, old wetlands that can never be restored? Is that possible, or can they always be restored?
Oh, no, Malcolm, there's some places that just can't be restored. You know, like they've been converted into something that's not possible to walk away from, right? You know, I think about, you know, infrastructure like airports, for example. So, you know, we have to be choosy about where we do these restoration actions. For sure, it can't happen everywhere.
Okay, thank you. And I'll insert a viewer question here that we have, and I'll read it out. Are there any is there any federal push to rehabilitate wetlands Australia-wide, like Blue Heart is doing on the Sunshine Coast?
I think the federal government is a signatory, or Australia is a signatory to the Ramsar Convention, which basically asks, wants to achieve the wise use of wetlands. That's at the key of the Ramsar Convention. So there is a lot of interest in the federal government to restore wetlands. And I think the commitment to including them in the greenhouse gas inventory, the commitment to the blue carbon method with the emission reduction fund, there's all sorts of signals that indicate that the federal government is serious about trying to support the restoration of Australian wetlands.
Thanks. I have another viewer question here. It says, are there any countries that Australia should be looking to as examples of best practice in how we shape our blue carbon policies? And if so, can you speak about what their approach looks like?
My goodness, that's a wonderful question. Indonesia was one of the first countries to put blue carbon up on the international stage. And they have made a national commitment to restoration. So, each country is really different with respect to the pressures on wetlands and also sort of like the processes that will work to restore them. But I really love the commitment by Indonesia to go forward. Mexico is also doing some wonderful work, and they one of their key lessons is the involvement of communities and Indigenous people in restoration. In Mexico, Indigenous groups can be very strong, and they write manifestos on restoration and their aim to restore their wetlands. So I think those two countries are, fabulous examples, and we could go on because in countries like Bangladesh and Pakistan, they have very strong government-led conservation and restoration programs.
Thanks for that. So we've all heard of sea level rise. So a related question there would be, as sea levels increase, do we expect to see more wetlands destroyed?
Well, I think we do stand to lose wetlands on low-lying wetlands that become inundated and cannot keep up with sea level rise. So they can't maintain their position over time. They will drown effectively. But there's also opportunity in sea level, with sea level rise. If we want to plan our landscapes to accommodate wetlands in the future, they will step back. Wetlands are, especially, mangroves and salt marshes, they're fantastic early colonizing ecosystems, right? And so, they can adapt if we give them space to grow, right? And that means, we need to think about those levee walls, where they are and whether it's useful for them to be there, or whether we should step them back to make sure that there's room for coastal wetlands in the future. I think if we eliminate coastal wetlands from the landscape, it's a huge travesty, and we stand to lose fish production and all manner of, services that we gain from them. So I think we really need, planning like I talked about in the Blue Heart, where we're sort of saying, okay, we know sea level rise is going to be an issue here. So we're going to make space in the landscape and take advantage of those ecosystems to help us adapt as best we can to climate change.
Mm-hmm. And in terms of the science, you mentioned carbon sinks several times there. In terms of the science, could you explain a little bit more about how the science of atmospheric carbon is stored in sediment? Is it in terms of timescales and capacity, is it similar to wood in trees, for example?
Well, sediment carbon stocks from mangroves have been around for thousands and thousands of years. We can still see them. So the evidence is that they hang around for a long period of time if they remain inundated, if they remain buried, if they remain often with the mangroves intact on top of them. For example, in Belize, you know, there's 10,000 years of mangrove root accumulation you can see on those islands of the Belizean Barrier Reef. So 10 meters of peat has accumulated through time, right? So the carbon stocks can stick around for a long time, but they need the environment to stick around for a long time. And if you know, dig them up and dry them out and drain them, then of course you have emissions. So if you reverse those conditions, then you're going to get storage. Basically, the carbon in organic matter can stay quite stable when it doesn't have any oxygen, and bacteria cannot break it down very easily.
Oh, that's fascinating. So you mentioned the dugongs there, and we all love dugongs. So could you tell us what's been done to protect dugong habitats?
Well, Helene, who gave the citation, is the very, you know, most expert in that. I mean, we have protection, of course, for dugongs. You know, we within the Great Barrier Reef Marine Park, you know, there's a lot of strong controls on what you can do. And in Moreton Bay, for example, we have things like go slow areas so that we don't have any boat strikes. So we try and minimize what happens to dugongs. But seagrass itself is subject to the impacts of climate change. So these are things that we're going to have to worry about more in the future. I mean, we have great protection in Australia, and we need to kind of work on that and build from that.
So I think that's probably all we have time for now, Cath. So thank you very much again, and congratulations again on this award.
Thank you very much. I'm deeply honoured. Thank you.
So we have to move on. And so to introduce our next awardee is we welcome Academy Foreign Secretary, Professor Francis Saporovich.
Thank you. I'm delighted to be here today to celebrate Academy honorific awardees, and in particular to celebrate the David Craik Medal for 2023. The David Craik Medal is a career award made in honour of the outstanding contribution to chemical research of the late Emeritus Professor David Craik. And in 2023, this medal was awarded to Professor David Craik. Before we hear David, I will read his official citation. Professor David Craik discovered a family of plant peptides called cyclotides and is a world leader in defining their structures, functions, and applications as eco-friendly pesticides and molecular scaffolds in drug design. He has shown how their unique structures makes them exceptionally stable and resistant to enzymes that would normally degrade peptide-based drugs. The work is significant because peptides are widely regarded as exciting drug leads, potentially safer and more effective than existing classes of drugs. However, previous peptide-based drugs are prone to instability and need to be injected, like insulin, rather than orally ingested. David's work on cyclotides shows how peptides can be stabilized and made more drug-like, thereby unleashing their potential in drug design. The natural function of cyclotides is to protect plants from insects, and his work has led to companies exploring cyclotides as pesticides. A cyclotide-based product, Xero-X, is now an approved eco-friendly pesticide for cotton and vegetable crops. Now let's hear from David.
Well, thank you very much. I'm really thrilled and humbled and delighted to have received the David Craik Medal for 2023, especially when I think about all the fantastic contributions that David Craik made to chemistry in Australia over the years, and also feel extremely humbled when I look at the past recipients of the award. What I'd like to do today is tell you a little bit about our work on the discovery and applications of some cyclic peptides that we call cyclotides. But it hasn't escaped my notice that there is some similarity between my name and that of David Craik. And it's not the first time that there's been such a similarity. And a number of years ago, I did some consulting work for a German pharmaceutical company who were actually paying quite well. And it wasn't until I received the contract via FedEx that I saw that they had addressed it to Professor Dr. Daniel Craik. And I'm not sure if the audience knows, but Daniel Craik is the actor that plays James Bond. So I'm completely confused as to how they could have made that mistake. But I do like my swim in the Brisbane River in the morning, so maybe there's some reason for that there. But the reason I put this slide up is because it's clear that James Bond, or 007, actually was interested in circular topologies very early on. And what I'm going to tell you about today is the circular topology of peptides. So the peptides that we work on are called cyclotides. And I should say that peptides are just small chains of amino acids. They're mini proteins. And we represent them with the one-letter amino acid codes, T for threonine, C for cysteine, etc. And what's unusual about the peptides that I'm going to talk to you about today is that rather than being a linear chain of amino acids, they're actually a ring of amino acids. And as is many instances of science, this work all started by accident when a Norwegian doctor went down to the Congo region in Africa in the 1960s and early 70s and found that when he was there, women would give birth very quickly when they went into labour. And it was because they were taking this plant here, Oldenlandia affinis, and making a tea from it, which they would then sip during labour, and that would accelerate the childbirth process. He called the molecule kalata kalata after the African word for fast, and took some of the plant back to Norway and discovered that the active ingredient was this molecule called kalata kalata. Nothing happened for 20 years or so until I did a sabbatical at Oxford University, and it was there that we discovered that this molecule had this unusual circular structure where, as I said, instead of being a chain of amino acids, it was a ring of amino acids. And furthermore, these cysteine or C residues there, which cross-link with each other, actually formed a knot in the middle of the molecule that you can see here with this embedded ring in the structure that's threaded by a third, what we call disulfide bond. And you can see that in the rotating model, where if you're an engineer designing a super stable structure, then you would love these cross braces and the interlocking cross braces. So when we did this structure, we realized this is why this molecule was so stable. It's a peptide that can be boiled and orally ingested, and no other peptides can do that. They either get broken down by the boiling process or chewed up by our digestive enzymes. So, for example, insulin, probably the most famous peptide, been around for 100 years or so as a therapeutic for diabetes. There's still no such thing as an insulin tablet, because if there was, the insulin would just be chewed up by our digestive enzymes. So it was really a breakthrough to find that we discovered a class of peptide that was super stable. This was my excuse to travel around the world and look at many plants to see if there are other examples. And to cut a long story short, there are many of these ultra stable cyclic peptides in plants. We've called them cyclotides, and they're, as I said, a very large family. They occur in all parts of plants, so in the leaf, the flower, the stems, in the bark, the roots. And you might say, well, why do plants make these molecules? In fact, we need to look at defense mechanisms. And if you're an animal and you're attacked by a predator, then typically what you can do is run away or fight or kick or scream. But plants can't do any of those things. So plants have evolved very good chemical defenses, ways of protecting themselves from predating insects. And so cyclotides are basically there as defense molecules. And we showed that very early on with some collaborative work with Marilyn Anderson, also a fellow of the Australian Academy of Science, where in Marilyn's lab, we took this cotton budworm, took the larvae from this caterpillar and fed it a diet containing cotton leaf. And if you do that, you'll find that over 16 days, the caterpillars grow to about 300 milligrams in size. But if you feed the caterpillars a diet containing cyclotides, they either don't grow or they die at a very early stage, at a first instar stage. And here's 3 controls and 3 treated or kalata-fed molecules. So early on, we knew that these cyclotides are defense molecules to protect insects, to protect plants against insect pests. We were able to understand how they do that, and working in collaboration with an Australian biotech company called Innovate Ag. They were developing a product based on this plant, Clitoria ternatia, or butterfly pea, that actually contains more than 70 cyclotides, and they've now produced a commercial product, which is an extract from that plant, that is now approved for use on cotton crops in Australia, along with vegetable crops. And the great thing about it is that it's peptide-based, so when it breaks down, it just breaks down to amino acids or food. So it's an eco-friendly next-generation pesticide based on these amazing molecules, which we call cyclotides. And I've indicated here that it's friendly for beneficial pollinating insects like bees. And this has been one of the problems with harsh chemical pesticides of the past. They've actually been harmful to some of these pollinators, which obviously has implications for the human food supply down the track. So we're very excited to work with Innovate Ag to get this molecule, or to get this extract developed into a product that's now, as I said, on the market and contains numerous of these cyclotide molecules. How do they work? Well, basically we know that cyclotides bind to the midgut membranes of caterpillars and basically cause disruption and make the insect feel very unwell and decide that it doesn't want to eat anymore. So here's an electron micrograph of a normal caterpillar, and here's one of a caterpillar that's been eating cyclotides, and you can see this swelling of those cells. Now, in the 10 minutes I've got, I don't have time to go through all the studies that we've done to confirm the mode of action, but suffice to say there's been 10 or 15 years of work trying to understand the mechanisms, and it's all to do with the unusual stability and shape and structures of these cyclotide-like molecules. And chemistry has played a vital role in making these molecules, in modifying them and re-engineering them. But I won't spend any more time on the agricultural applications for the moment, because I'd like to now swap to pharmaceutical applications of these molecules. And as I've indicated in the slide here, our agricultural applications are mainly based on the natural cyclotides. We take the cyclotides from plants and we deploy them for agricultural applications. But we also know, because of this exceptional stability of cyclotides, maybe we could solve some of the problems with peptide-based therapeutics and make some super stable therapeutic peptides. And that's what I'm going to spend the rest of the talk on. And rather than go through each individual story where we've looked at lots of examples, I'll just give you the principle, where what we've done is we've taken a super stable cyclotide, a peptide epitope that may have been discovered in a pharmaceutical company or in an academic lab that has a therapeutic application, and we can graft that epitope onto a cyclotide framework to make it stable. And we've now done this in many, many cases, and looked at a whole range of different targets, from enzymes in our bloodstream through to receptors on the surfaces of cells, through to intracellular enzymes, through to protein-protein interactions that are implicated in cancer. And so far, there's one molecule that's gone into clinical trials through a Swedish company for multiple sclerosis, and I'll come back to that at the end of the talk. But we're very hopeful that there'll be some other applications coming out over the next couple of years of these molecules as therapeutics. So in preparation for that, we want to work out how to make these peptide-based therapeutics, which are very specific and therefore should have fewer side effects than traditional drugs. How can we make them inexpensively? And I guess this is where we came across the idea that if plants make these molecules naturally, then maybe we could adapt the plants as biofactories for making therapeutics. And so the idea behind this is that we have our plant that produces the DNA that encodes for the peptides. We come along as drug designers and decide that we want to graft something onto that drug and re-engineer it. We can just reverse translate, make a modified gene, which we can then put back into a crop plant to produce these peptide-based therapeutics in a very sustainable, eco-friendly way. You just need some light, sunlight and water and nutrients for growing these things. And we could put it into a crop plant and express the peptides in the leaf of, say, tobacco or soybean, harvest the plant, refine the pharmaceutical ingredient, and then put it into a tablet or an injectable, whatever the product would be. So we're in the process of doing that. But even more exciting from my point of view is that we could also think about putting the therapeutic design a peptide into an edible part of a plant so that you could actually eat your medicine as part of your diet. Sounds far-fetched, but we actually made fantastic progress in along those lines. And we've done this in a range of different host plants, from Arabidopsis, which is the test plant, through to tobacco, petunias, and a whole range of other plants that we're working on at the moment. We realise that there are many things to consider. How do we deal with overdose, the regulatory process, but all those things are things that we're working through. And we're applying this for applications in cancer and pain, and as I mentioned, multiple sclerosis. Here's just one example of an agent that involves a peptide that blocks a part of the blood coagulation cascade, where we've produced a plasmin inhibitor in plants, shown that it's identical to the chemically produced molecule and has super potent activity in an enzyme assay. The one that I'm most excited about, though, is our tomato work, where we've actually produced a drug lead for multiple sclerosis in the fruits of tomato plants. And this is work by a PhD student, Lynn, shown at the bottom here. And what Lynn's done is produced this therapeutic lead molecule in tomato fruits. Now, if you don't like eating your Greek salads to solve your multiple sclerosis, perhaps you could have the tomato juice, or even the Bloody Mary in some cases, as a way of delivering these therapeutic peptides. So sounds very far-fetched and pie in the sky, but I think it's taking us back 2,500 years to Hippocrates' time, who said basically, let thy food be thy medicine. And now I think we're turning that into a reality. So I have limited time. I'd just like to finish there by saying I hope I've given you an impression of how we can go from discovering peptides and interesting molecules in plants, determining their structures, deciphering their functions, and then re-engineering them to graft them with new activities as therapeutics. And so effectively we've come the full circle. And if I can go back to the James Bond analogy with the circles, James Bond was licensed to kill, and I hope I've convinced you that we're licensed to pill. So I'll leave things there, but of course I didn't do any of this work. This was all done by a brilliant team of postdoctoral fellows and research assistants and students, and their names were on some of the slides as we went through. So I'd just like to acknowledge their fantastic contributions, along with a large number of funding bodies, from philanthropic agencies through to the national science funding agencies, but also to acknowledge the Australian Research Council Centre of Excellence for Innovations in Peptide and Protein Science, which is really a flagship program for doing this sort of work. So thanks to all those people, and it's fantastic to talk to you today. And thank you so much again to the awards committee for this award, which is really greatly appreciated. Thank you.
Thank you, and it's now my absolute pleasure to welcome David Craik live with us online. Welcome, David, and congratulations on this award.
Thank you very much, Malcolm. It's a pleasure to be here. Thank you for having me.
And now we'll go to some questions, and I'll just remind the online audience that you can ask David a question by following the QR code on your screen. But I'll move to a few questions, and I very much like let food be thy medicine. So in that area, let me ask, if one, say, has a tomato as a food, are you worried about processes that happen to food that might damage that efficacy? Are you bruising or rotting before someone eats it? Is that a relevant issue?
Yeah, definitely, Malcolm. I mean, I think it's the same sort of issue as we would expect for our regular food. We want to make sure that it's stored and delivered in top conditions. So there's really no difference in that regard. A bruised tomato wouldn't be very attractive to eat anyway, so you wouldn't get your medicine. So we need to make sure that these things are delivered and transported in a healthy, fit for consumption state.
Yeah. Okay. So in terms of the general approach, you described it as far-fetched, but obviously it isn't. Taking medicine via food compared to a pill or an injection, what would you say are the advantages of that?
Yeah, well, look, I should say at the beginning that this is not for every medicine that we'll take. There's probably a niche set of medicines that we'll do this with, but it's really a disease-specific indication. So, for example, someone with inflammatory bowel disease, that has pain associated with that might like to have a food as part of their diet that could relieve the pain or relieve the symptoms. So for chronic diseases where people are having to take their medicines for the rest of their life, if there could be some relief from either the pain or the condition itself, I think that's an advantage. It saves them having to take up, lots and lots of tablets or injections. So there's the convenience aspect, and there's also to some extent a patient compliance aspect. Sadly, my father passed away a few years ago, and he had about a dozen tablets every day he was taking, and sometimes he'd get confused and not be quite sure whether he'd taken Monday, Tuesday, Wednesdays, or but he would never forget his cup of tea at 10 o'clock in the morning. And if he could have had a cup of tea that might have had a medicine, or a few of those medicines in it, I think his compliance for medicines would have been better. So there's the convenience, there's the compliance issue. In terms of developing countries, there's also an issue there where sometimes a lot of our current medicines might need a cold chain storage, like antibodies, for example, need to be refrigerated. If people in developing countries could be growing medicines locally that were super stable like these ones, then we could get more high-tech medicines out into the developing world. But also the sustainability issue, because if we're producing medicines in plants, then as I mentioned in the video, we're just using sunlight and land and some water and some nutrients, of course, and the labour to do it. But that's potentially more sustainable than chemical manufacture. It's been estimated that for peptide synthesis, 1 kilo of a pharmaceutical ingredient actually is associated with up to 3 tons of waste. And so it's a much more sustainable process if we can use green methods to produce some of these molecules. So I think there's lots of potential benefits, but as I said, to emphasise, it's not going to be for every medicine and not for every food. It'll be a case-by-case basis.
Yeah, that's fascinating. So you didn't mention dosage. I wonder about dosage. So how do you control dosage? Say, if you're eating a tomato, do you have to eat so many? And can that be affected by, let's say, the age of the tomatoes? Do we have to test food before you eat medicine-enhanced food?
Yeah, all very, very good questions and things that we've thought about. So again, this technology won't be for every medicine, and it certainly wouldn't be for medicines where there was a very tight margin on the doses that you needed. But for example, we have one peptide that's about 150 times more potent than morphine as a painkiller, and it's only required in very low doses, and it's a very, very safe molecule. So having something like that as a painkiller in a food that would provide a background level of pain relief, where it wouldn't matter if you ate 2 tomatoes rather than 1, would be the sort of application. We wouldn't do it for a blood pressure medication or something where there needed to be an accurately controlled dose. But it is certainly one of the issues that we're thinking through. Another example we've got is actually an anti-obesity molecule, and we're exploring that at the moment. And my dream would be to put that into potatoes, so you could have your French fries and not worry about obesity. But there we've actually got an inbuilt advantage, because this molecule is actually an appetite suppression. So you couldn't eat enough potatoes, you would lose your appetite for potatoes as you took the medicine. So we need to think about these almost self-regulating negative feedback mechanisms in the medicine. So there's lots of interesting things we're doing there. The other one that we've been thinking about is if some of these were delivered in medicinal teas, as was the original concept that we got this from with the ladies in Africa. We could think about brewing a tea and then developing some litmus paper test where you could dip a sensor, in the form of a litmus paper type test into the tea. And if it turns green, then you know that the dose is right. If it's not green, then you can let it brew for a little bit longer. So that would be a high-tech sensor, hopefully very cheap in a litmus paper format, that could look at dosage. But it's definitely an issue, and definitely not something that means that this technology will be applicable to every sort of medicine. But I think there's a place for it in some cases.
That's fascinating. So let me ask perhaps maybe it's a simpler question. So you mentioned about the circular shape. What shape do peptides often take? And in your average plant, what percentage of peptides would there be in the circular shape?
Well, cyclic peptides are a rarity in nature, or at least we haven't discovered very many of them so far. I think there's actually more of them out there than what's been discovered so far. But just let me clarify this circular argument. Peptides have got many different shapes. Peptides are just chains of amino acids, and the code of amino acids, the sequence of amino acids, determines the three-dimensional shape. And so they actually fold up into a compact shape, even if they're a linear peptide. And I've got a little model here. So this is a typical peptide of 30 amino acids. It folds up into quite a complex shape. And it's this three-dimensional shape that determines the function of that molecule. What's different about our peptides is that their chain of amino acids joins together at the end, but then it still folds up into a complex shape. So my other model here that shows that circular backbone, you can see the red chain of amino acids has no ends, they're joined together. And then we have these cross-linking disulfide bonds that tie the whole thing together. And it's this compact shape that really determines the activity of the molecule. And so there's an infinite number of possible shapes of these molecules, and each different shape binds to a different receptor. And what we do is we try to engineer a little bit of peptide sequence onto one of these loops to hit a particular receptor very, very potently and very specifically. So the short answer to your question is peptides have many different shapes, mostly linear peptides that fold up. But when you join the ends, what you get is an extra bonus in stability. And that's why we can turn these molecules into therapeutics.
Okay, that's it. I think you've answered my next question there by telling us the function of the shape. But we actually have some audience questions, and I'll go to those. So one of your slides briefly mentioned public perception of genetic modification in food. Do you expect this to be a major barrier to the adoption of your technology? And do you have any thoughts about how to change that public perception?
Yeah, look, that's a great question, and obviously there are sensitivities on genetically modified organisms in different countries. It's a very regional thing. It's a very much accepted technology in the US, and very much concerns for people in Europe. So Europe wouldn't be the first place that we would be taking this technology to. But I think one of the initial problems with genetically modified technologies is that it was not explained very well and people couldn't see the benefit to them. But I think if people see that there's a benefit to having an edible or genetically modified plant that will give them a health benefit, then I think, and that the risks are carefully evaluated. And I should say that genetically modified organisms are very highly regulated areas, so all this work would need to be done under very strictly regulated conditions. So I think ultimately if the technology proves beneficial to people, then I think that will draw people's acceptance of the technology along. But it's obviously important as scientists that we try to explain the technology so that people's concern about it are minimised.
Thank you. We have another audience question. It's actually from your introducer, Francis Zaporovich, and she asks the stability of cyclotides. Cyclotides allows for oral administration, but do you also break down in the body, or do they also break down in the body, or are they removed in their native state?
Yeah, thanks, Francis. Yeah, so you're correct that some cyclotides are orally active, and that's a really big deal for peptides, because generally peptides are not orally active. I mean, insulin, probably the most famous peptide, has been around for 100 years, but there's still no such thing as an insulin tablet because our body would simply chew it up. It would assume it's another peptide or protein and chew it up. But the thing about cyclotides is because they have that cyclic backbone, they're stable. So they do survive the gut, they get into our system and can act as a therapeutic, but then they're excreted very often in their intact state, and then they break down in the environment. So not all cyclotides are orally active, but a number of them are, and we're trying to more fully understand what makes something orally active, because people like to ingest things rather than inject things. So we think if we could make an orally active insulin, that would be a really important breakthrough for diabetics, so that they wouldn't have to inject themselves very regularly.
Thank you, David. I'm sure we could carry on with questions, I think, but we're out of time, unfortunately. So let me again thank you for being here today, and congratulations again on your award.
Thank you very much. I feel very honoured and humbled to receive it. Thank you very much.
Thank you. So now we'll move on, and to introduce our final awardee of today is again Professor Francis Zaporovich.
Thank you. I'm delighted to be here to celebrate the Thomas Ranken Lyle Medal for 2023. The Thomas Ranken Lyle Medal is a career award that commemorates the contribution of Sir Thomas Ranken Lyle to Australian science and industry generally, and in particular to his own fields of physics and mathematics. The purpose of the medal is to recognise outstanding achievement by a scientist in Australia for research in mathematics or physics. In 2023, this medal was awarded jointly to Professor Susan Scott and Professor Nick Wilmot. We will now hear from Susan, but before we do, I will read her official citation. Professor Susan Scott is an internationally recognised mathematical physicist who has made fundamental advances in our understanding of the fabric of space-time in general relativity and in gravitational wave science. Her groundbreaking discoveries probe the existence and nature of singularities and the global structure of space-time and possible initial and final end states for cosmological models representing our universe. Susan has also been a pioneer in the analysis of astrophysical signatures in gravitational wave experiments, including the searches for gravitational waves from asymmetric neutron stars and from in-spiraling binary systems of black holes and neutron stars. She has played an important role in the development and promotion of gravitational research worldwide, and a leading role in Australia's participation in the first direct detection of gravitational waves in 2015. Now let's hear from Susan.
Firstly, I'd like to thank the Australian Academy of Science for awarding me the Thomas Ranken Lyle Medal for 2023. It really is a very great honour. My talk is going to cover two key aspects of my research, and both of those are a matter of gravity. When I was a child, I was amazed to watch the first landing on the moon by the astronauts. And as I watched them leaping above the lunar surface, I was totally mesmerized. And I knew that I was excited by gravity, and I think this ignited my lifelong interest in gravity. Of course, the great modern theory of gravity is general relativity theory, and that was proposed by the famous physicist Albert Einstein, who is also responsible for many other great contributions like the photoelectric effect, Brownian motion, and special relativity. But I like to think that the general theory of relativity is his greatest contribution. And there are two great predictions which come from his theory, and one of them is the existence of black holes, and the other one, of course, is the existence of gravitational waves. And these will incorporate the two key parts of my talk. So the first one, black holes. Well, in 2020, Roger Penrose received half of the Nobel Prize in Physics. And he did this for a very extraordinary piece of work on black holes. And this was the famous singularity theorem of 1965. And that theorem was revolutionary. Basically, it assumed three main components: an energy condition on the matter in the space-time, a causality condition on the space-time, and also a trapping condition. And this means that somewhere gravity is strong enough to actually trap a region of the space-time. And under these very broad assumptions, the singularity theorem predicts the existence of a singularity. And this was an amazing breakthrough because it showed that the singularities were not purely an artifact of the symmetries in the models that were used to look at these situations. It showed that they were a general feature of general relativity coming from the theory, and therefore, you know, they should exist. Black holes should exist exist throughout the universe. And this preceded by a number of years the first serious candidate for a black hole being identified by astronomers in 1971. But the thing is, the singularity theorems are actually incomplete. What they do is they predict what we call an incomplete causal geodesic or curve. And those are the sort of curves that you and I move on, that observers move on. And it means that somehow these curves come to an abrupt end. But it doesn't actually predict a singularity. But what do these incomplete causal curves begin or end at? Is it a singularity, in fact? And if so, is it a curvature singularity where the curvature of space-time blows up? And where is it located? So these remaining questions from the singularity theorems have been a major quest in general relativity for the last 50 years. And my work in this area has been involved in trying to solve these questions. So in particular, I've studied a lot of aspects of singularities of space-time, their properties, how these singularities occur, and what different types we have of them. This led me to develop a new, what we call boundary construction for space-time. This is putting an edge on space-time where all these nasty singularities occur. And in fact, our boundary is so general, it's called the abstract boundary. It incorporates things like the singularities, of course, but also regular points where we can extend the space-time, and also regions at infinity. And in the diagram, we see what we call the Penrose diagram of the black hole. And the diagram to the right is like the main part of the universe. And at the edge of that, we have regions at infinity on the edge, on the boundary. Then we have a horizon through which observers can pass. And those are regular points on the edge of that particular piece of space-time. And they can proceed through to the sort of wiggly line at the top, which is actually the curvature singularity. And you do not want to end up there. So where do the incomplete causal curves begin or end? Well, we proved a general result, myself and Ben Whale, in 2021 called the endpoint theorem. And what this very general theorem does is it provides an endpoint for these incomplete curves predicted by Penrose's theorem. So it gives a place where wherever they end is happening. We've also done some further major work on this problem, and we've shown that these incomplete curves actually end at what we call an essential singularity. And that means it is some kind of singularity they end at. We haven't specified there, we haven't been able to at this point, but we show that these curves actually end at some kind of singularity. So what we've shown is that there isn't a place where these incomplete curves of Penrose's theorem go. They end at some kind of singularity. And what remains to be shown, and that's something we work on now, is that these predicted singularities are actually curvature singularities where the curvature of space-time blows up, like at the center of a black hole, as you see in the left-hand diagram, and also on the necklace that I'm wearing, but also at the beginning of the universe in cosmology at the Big Bang. So this is a work in progress, but we are a long way down the track. The other area that I work on is another prediction of Einstein's theory, namely gravitational waves. And these were predicted by Einstein himself in 1915. And basically the idea is that all moving masses in space-time jiggle space-time around them. And in so doing, they generate gravitational waves or ripples or distortions in space-time, which travel outwards at the speed of light. So thing to remember is they're not part of the electromagnetic spectrum. They're a different spectrum, but like light, they also come in different wavelengths. There are currently 4 ground-based gravitational wave detectors around the world, 2 in America, the LIGO instruments, 1 in Louisiana and 1 in Washington State. And each of those has 4-kilometre arm lengths. So they're very big instruments. There's also another 2, 1 in Europe called Virgo and 1 underground in Kamioka in Japan. And both of these 2 instruments have 3-kilometre arm length. So the LIGO and Virgo instruments started being constructed in the early 90s. And at that time in Australia, we already had endeavours in developing components of the interferometers of these detectors underway. But in 1997, I decided that we needed to start doing the actual gravitational wave science that we could expect to look at and discover using these instruments. So in 1999, I established a formal collaboration with LIGO in data analysis. And during these years, we contributed key components to the LIGO data analysis system. We also pioneered exchange of seismometer, power monitor and magnetometer data from the various observatories. We looked at the best ways to situate a global network of gravitational wave detectors. We looked at effects of gravitational lensing on gravitational waves. And we actually designed and led the first Australian search for gravitational waves from Cassiopeia A. So we were very busy in all these years leading up to detection. And finally, on the 14th of September 2015, we made first detection of gravitational waves, and it was from two black holes, 36 and 29 solar masses, spinning around each other and then coalescing to form a bigger black hole of 62 solar masses. And this happened 1.3 billion years ago. And you can see the two signals at the bottom from the two LIGO detectors. So we had finally discovery of gravitational waves, and we fully participated in it, including the science as well. And we celebrated this at Parliament House with the Australian team in early 2016. So what did we learn from it? Well, this was the first direct detection of a gravitational wave on Earth. It was the first detection of a black hole binary system. It was the first observation of black holes merging. And from my point of view, and most importantly, the first test of general relativity where the gravitational field is very strong. Because up until that point, we had not been able to test the theory in those regimes, and it stood up remarkably well. 2 years later, we had the first detection of a binary neutron star system merger. And this was a spectacular event, the first such observation, in fact. And it was the first direct evidence of a link between these neutron star mergers and short gamma ray bursts, which had been a mystery for about half a century. And subsequently to us detecting the wave, the astronomers actually found the source and were able to image it across the electromagnetic spectrum. So we got an amazing amount of science out of that one event. Since then, we've had announced over 90 gravitational wave detections from binary systems of black holes and neutron stars. The blue ones up the top are the binary black hole systems. The vertical line, two smaller ones, creating a larger one at the top. Down the bottom of the diagram, we have some orange ones. They're the neutron stars. And in between 2 and 5 solar masses, in our what we call the mass gap, we have some ambiguous items, either a very heavy neutron star or a light black hole. Well, this is just the beginning of the field, and we go on to look at the lives of these binary black hole systems, but we're also wanting to find things like gravitational waves from the explosion of a supernova to try to understand how that process works. We also want to detect the continuous gravitational wave stream from an isolated neutron star with a small asymmetry, like a tiny mountain. And this is very, very important because one of the big mysteries of physics and astrophysics is what is a neutron star made of? And we believe gravitational waves will help unlock that secret. And of course, we do expect the unexpected. We can call a gravitational wave signal that we don't know what it is. And of course, that's a very exciting prospect. So huge endeavours and discoveries lying ahead. Finally, I would like to thank the Australian Research Council for their continued funding of our projects and activities over many years. And to thank you all for joining me here today for this talk. It's been a great pleasure. And as I said at the beginning, a very great honour. Thank you.
And now it's my great pleasure to welcome live online here Professor Suzanne Scott. Welcome, Susan, and congratulations on your award.
Thank you very much, Malcolm. As I said, it really is a tremendous honour.
And I'll just remind the audience again that you can ask Susan questions using the QR code in the usual way. But I'll kick it off with some questions if I may. So I'll ask one for the non-physicists in the audience, and I'm sure there are some. Could you explain further what you mean by the singularity theorem predicts the existence of a singularity? And what exactly is a singularity?
Okay, well, singularities are a great thing in general relativity. They're actually places in space-time where very strange things happen and the laws of physics break down. So an example that you all know about is that coming from the center of a black hole where the curvature of the space-time goes infinite. So this means that we've had the finite mass of a star squashed into a single point effectively. But there are different kinds of singularities, and we're not even sure what all the different kinds are. But another example is if, for example, space-time pinches off at the edge. And this means that an observer like us going to the edge, traveling to the edge, will suddenly blip out of existence. So we don't, there's no sort of big physical effect there like infinite curvature, but it means that there's some sort of problem. So these are the singularities of space-time, and that's what the singularity theorems are about.
That's amazing. Okay, so your work on the famous Penrose-Hawking theorem, singularity theorem, what does it tell us about black holes and the Big Bang, or does it tell us about the Big Bang?
Well, you know, the Penrose and the Hawking singularity theorems from the 1960s are incredibly important in general relativity theory. So they start with very general assumptions on a space-time, like an assumption of an energy condition on the matter in the space-time, a causality condition on space-time, and a trapping condition, which is that some part of space-time has sufficiently high gravity that it traps that particular region. And that, of course, occurs, for instance, when a star is collapsing. So what they show is that under these very general conditions, there exists in the space-time an incomplete causal curve. And that's simply a curve or a world line that observers like yourself and myself and photons and so on travel on. And it means that somehow they blip out of existence at a finite time. So the singularity theorems show that we have these incomplete curves, but for the last 60 years, it's been a mystery as to what happens with these curves. And the big questions are, where do they actually go to? And secondly, you know, an observer traveling along one of them, what are the physical conditions they encounter? For instance, do they actually approach infinite curvature? And so the work which I've been engaged with various people over the years is to try to what we call complete the singularity theorems. And we've completed 2 out of the 3 big steps for these theorems. We've shown a location where these incomplete curves that we travel on actually end at the edge of space-time. And we've shown that they end at an actual singularity of some sort, and that the observer blips out of existence at that point. So this is a very long way down the track towards completing those theorems. And what remains to be shown is that the observer actually approaches a place of infinite curvature, such as at the center of black holes or at the Big Bang. So that's the final step, and that's where we're going at the moment.
Okay, that brings me nicely on to the next question. And I don't know if it's particularly well posed, but you mentioned getting close to the singularity, and there are different areas where you could go and places where you don't want to go. Do you think in the future it would ever be possible, even conceivably, to send a machine into a black hole and see what's there and send information back? Could we even get information back?
Well, I think it's possible in the future that we can send a machine into a black hole. But as for the rest of it, I mean, I think the answer is certainly no. I mean, for a start, as the machine is approaching the black hole and approaching the event horizon, it will be what we call spaghettified. And that means it will be drawn out into sort of vertical filaments of single atoms. So it'll be completely broken up and would not be able to perform any function beyond that. But even if it could go beyond the horizon, the thing about a black hole is that no information can actually escape from the event horizon of a black hole. So even if it could collect information, it would not be able to transmit it back to us. And there is a possibility you could think about a black hole as a type of wormhole. So the machine would go into the black hole and be flung out the other side by what we call a white hole. But the thing is, these wormholes are very unstable. They're transient and pinch off as any object would try to sort of pass through them. So the answer is no.
Okay, that's clear. So you mentioned, you mentioned in your talk about using gravity waves to look into a neutron star to see what it's made of. How would that work? How would you tell what it's made of from the waves that are emitted?
Yes, well, the first point is that we have not detected waves from a single isolated rotating neutron star as yet, because they're somewhat weaker than the sort of waves we've been detecting. But it is very much on our agenda for the future. And they generate these waves by a very small asymmetry. They may have like a millimeter-sized mountain, not very small, but in a dense object like that, it makes a difference. And if we can detect this continuous stream of waves, then what we can do is infer macroscopic properties about the neutron star, such as its moments of inertia, ellipticity, magnetic dipole moment. And we can use those to strongly constrain the type of matter inside the neutron star, in the core of the neutron star, because at the moment, we don't really know what the core of a neutron star is made of. And by looking at, for instance, the amount of deformability of that star via its ellipticity, then this will help us to constrain the type of matter. And you know, there are many possibilities, like sort of basically it's neutrons with some protons and electrons, or it could be much more exotic forms of matter like strange quarks and so on. So yes, we're very anxious to make this sort of detection.
And you showed in the video about two black holes colliding, of course, which created the gravitational waves that were detected. Could we detect other activity or other nucleus of gravitational waves from other events? Is it just that those are the largest that as our detection ability improves, are we going to detect them from other celestial issues and use that for extra data?
Yes, that's a good question. And I think the thing that most people don't realize is that any kind of moving masses that are accelerating and are somewhat asymmetric actually produce gravitational waves. So, for instance, a human waving an arm around produces a gravitational wave. But the thing is that is incredibly weak. So weak that we can't feel it passing through us. And certainly we can't detect it with our gravitational wave detectors. And something like a system of two stellar mass black holes rotating around one another and then smashing together, what we've got there is two very dense objects, so sort of infinitely dense. The system is very asymmetric as they get close together. And before they coalesce, they're traveling at about half the speed of light. So they produce very strong gravitational waves. But, you know, they're not the only source that we can detect. We have already detected gravitational waves from a pair of neutron stars colliding, which was a very spectacular event. And they're not as dense as black holes, but they still produce quite strong gravitational waves. And looking to the future, we have a number of sources that are on our horizon as we get more sensitive instruments. We'd like to detect the gravitational waves from a not too far away supernova going off. Also, the gravitational waves, as I've mentioned, from an isolated rotating neutron star, which is a little bit asymmetric. We're very keen to get hold of those, but also, you know, lots of other possibilities, like from potential dark matter sources, and also information about the very early universe.
That's amazing. I'll just try and slip one question in at the end before we've got time. What is it that limits the detection then? You mentioned about precision. Is it the length of the arms, or is it data processing techniques, or there are other factors?
Well, yeah, I mean, predominantly it is, of course, the detectors. I mean, the next generation of detectors, the third generation planned for the mid-30s, we're thinking about an instrument that goes from 4 kilometre arm length to 40 kilometre arm length, which is a really large instrument, and that will give us, a factor of 10. And that makes a huge difference to the volume of universe that we can collect events from, but also the possibility of, detecting much more weaker waves like the ones from supernovae and isolated neutron stars.
That makes a lot of sense. Okay, I'm afraid we have run out of time. We could carry on chatting, but thank you very much again for that. And let me congratulate you again on this award.
Thank you so much, Malcolm. It's been a great pleasure, and I do feel very honoured. Thank you.
Great, wonderful to talk to you. So I'm afraid that's all the questions we have time for. And so we thank Susan again. And it brings us to the end of our event this afternoon. So I'd like to thank everyone across Australia and the world who has joined us to celebrate Australian science and the work of our extraordinary awardees. And I think you'll all agree with me, they are extraordinary. Next week, on Thursday, the 31st of August, we will be hearing from another 5 of our career honorific awardees from 2022 and 2023. More information is available on the Academy website. So we hope you will again join us to celebrate excellence in Australian science. I'm Malcolm Sandbridge. Good afternoon.